Okay, good morning everyone, and this is Raj, and I will just go over this. It's a summary report of the project 1, which is basically, as you know, we have divided up this project into two, project A and B, based on the cell lines, which are classified as TET on or TET off. So this project is to summarize the results from the first series of TET on cell lines, which were 18 cell lines, which we have received, and perform this microtumor testing with those cell lines. And I'll just briefly go over the protocol. So this is the second slide. So after we received the cell lines, which were fine, and they came on dry ice.
I'm sorry, Raj. I'm sorry. I don't think we can see the second slide.
Yeah, not yet.
Oh, because I'm switching.
We basically see your share screen on your desktop.
Where did it go? Did I disconnect?
So if you can shut the share screen off, then we'll go back to the main slide.
Yeah, I think I somehow got disconnected from WebEx.
Well, right now I noticed a change on your desktop, so maybe you're reconnected.
So I'm still connected?
This isn't all of it, I hope.
Yeah, we can still all see your desktop. So all you have to do is advance to slide 2. So I see a PDF and it still says page 1.
Okay. Do you see now 2? Number 2 slide?
No, I think it stopped again.
Oh.
Okay,
There we go.
now it works.
Now you got it.
Works.
So now you see it, slide 2?
Yes.
Yep. So somehow it's not showing me that I'm connected to WebEx. I don't know why. So let me make sure. So this is slide 2, and I move to slide 3. Does it happen on your screen?
We're on culture mode.
Yeah, it's on the culture protocol now.
Oh. So it's showing me, I'm showing it says you are sharing your desktop, and at the bottom, so I don't know why it's not showing.
It could be an internet issue, maybe, but see, it works on and off, so I noticed we changed something right now, and now it's frozen again. So maybe you could go back to
Slide 1.
Slide 1? Okay. So you want me to restart the presentation?
That would be great.
Okay.
Well, as long as you can change slides and move. If it doesn't work changing the slides, maybe you could send the slides out and we could just walk through them with you.
Or alternatively, Amit can just move the slides and you can just say next slide. So Amit, you can take the ball back.
Right.
Okay, Amit, why don't you take control and let me go through the slides?
Sure, just give me 1 minute.
So let me stop sharing. I'll push that button. So I'm back.
Okay, so I'm going to move to the second slide.
Okay.
Which is where you stop.
Okay, everybody sees it?
Yeah.
Yes.
Okay,
Yeah.
good. So let Amit move the slides so I can just go over them.
Sure.
So as I was mentioning that we received the TET-on slides, cell lines, and we thought them, placed them in the culture flask as recommended by the protocol we received. And we divided them into two groups. First set was A549 and H1703 cell lines, and the second was HT29. And that was based on our experience that the HT29 slightly grows faster than the other cell lines. And we grew these cells to 70 to 80% confluency, and then we harvested them using the XXX, and in this case we didn't passage them, we just used them, and we counted the cells using a automated cell counter, and we call that day 0. And now, after we got enough cells, and we calculated that they are enough to prepare microtumor beads, we went to the next step, which is microtumor production. So next slide, please. So to make microtumors, this is a brief protocol. We use a high density, we call high density Hugh biogel because it forms a gel. It's a 3 milligram per ml concentration we use, and generally we neutralize it because we are putting live cells in it. So it's around pH 7.5 and ready to use for making the 3D beads. So typically we count enough cells per experiments and centrifuge them down. Then we just resuspend them in the FPS, which was supplied with the Pfizer protocol. We use the same FPS. And our calculation is that we put 10,000 cells in a 10 microliter Hugh biogel bead. That's where our starting point. So each microtumor will have 10,000 cells in a 10 microliter volume. So after mixing the cells in our gel, we dispense them in our customized this microarray we have created here, which allows us to make 96 microtumors same time. So there are 10 microliter mixture of cells and Hugh biogel dispensed on this microarray, and it takes about 7-8 minutes. Can you go back, please, Amit?
Yeah, sure.
Okay. So 7-8 minutes, the gels will polymerize, and then we transfer all these microtumor beads directly into assay plates. So these are 96 cell plates with media.
Just to try to get an understanding, in each of these 96 wells, how many beads and how many cells on a bead? So, you know, how are these cultures start on numbers?
So there are 10,000 cells in each bead to begin with, and there is one bead in each well.
Okay, so you're starting with 10,000 cells on one bead.
Exactly. Inside the bead. Yeah, they're mixed in the bead, in the gel bead.
Okay, thank you.
Yes, so we mix the gel and cells together, then we create the bead. And once it polymerizes, we transfer them to assay plate. So there is a 1 microtumor bead per well. And as you'll see from the image, they are about 2 millimeter size. So there is a 1 bead in each well, and they are suspended in this complete media. And we start adding the DOX media every third day. We change the media, and just like feeding the cells with fresh media. Next slide.
Sure.
Okay. So we started this 21-day assay for this particular project. So on day 1, that's our like control day. Day 1, day 7, day 14, and day 27. We perform these assay points. So we take the live cell imaging. We perform on 1 bead or 2 beads. So they are triplicates for each conditions at least. So on one set of microtumors, we perform this calcium AM staining. We stain them with calcium AM, shown here, microscopy protocol. That we remove 2 microtumors and we add calcium AM, incubate it, and take image at 4x and 10x and record those images. And this gives us the total viability, as you'll see in the images, the total green fluorescence you see. At the same time, we perform MTT assay on the same conditions, like 3 or 4 microtumors for each conditions. And we perform the standard MTT assay shown here from day 1 to 21. And then we take the mean and place the ST and plot them those graphs. We also collected about 5 tumors from those each conditions, and we just put them in -80 freezer, and we'll be shipping them to your place for your analysis for DNA or RNA.
Raj, so
yeah.
the microtumor, except from one bead, or one bead you could form multiple multitumor?
No, it's one tumor. Microtumor is one bead.
One bead. Okay, so you had to.
When I say microtumor, it is one bead.
Okay, got you.
So we either call them tumor bead or microtumor.
Mm-hmm.
But there is one in each well, and the numbers I'm showing here, like 2 for imaging, 5 for freezing, about 4 or 5 for MTTSA.
Okay.
So this is the summary of all the like the protocol and readouts. And next set of slides are individual experiments showing the calcineurin imaging and MTTSA. Next, Amit. So here's the first set, A549-4029NT, and on the bar graph is showing the MTTSA on day 1, 7, 14, and 21, and corresponding images at 4x and 10x on those days. These microtumor beads are stained with calcineurin. So this just shows the total viability of cells as they are growing over a period of time. And as you can see from day 1, day 7, 8, there is a larger and larger these colonies forming. And everything is formed inside this huboidal bead. Cells never come out. You will not see anything in the plate. Everything is going inside this bead in suspension.
This is being stained with what dye did you say?
Calcineurin.
Calcineurin, which is a mitochondrial live cell imaging.
And so the fact that the center is a little bit darker, is that really reflective of are they becoming hypoxic like a normal tumor does, or?
Yeah, at the end at 21, you see like little darker areas that could be hypoxic conditions inside.
Do you believe that's a real result, not just an artifact of the staining protocol itself?
We have seen, like if you let this tumor grow maybe another week, then you'll see a very discrete hypoxic center.
Okay.
Yeah, we have seen on some images you can see it very clearly that there's a hypoxic core in the middle.
And have you used the hypoxy probes to really define the hypoxic area?
But yeah, we have done a number of experiments where we stain with like the HIF or hypoxic specific biomarker.
All right, thanks.
Hi Raj, this is Andreas. Is this optimized time that you have the calcine in the tumor, in the microbeads?
Yeah, this is a very standard 20-minute staining.
Okay.
Yeah, so we just take out microtumor from on these particular days and stain them with a standard calcineurin staining protocol. It's fixed protocol.
All right.
And
thank
these
you.
images are taken one time. We don't use these microtumors for any other analysis.
Yep.
So this is one example, and if you go through next slide, Amit.
Are these docs treated or non-docs treated?
These are docs treated.
Okay, so these are all docs induced. Okay.
Every third day we add a docs media throughout the experiment. Yeah. So here is the next one, DXX9, and keep going.
How do we distinguish calcium from green?
I forgot. The calcineurin is green, right?
That is correct.
Yeah. It stains. The cells are green, and
you can see the difference between that and the green of the GFP.
Not in this one, probably not.
We should not use calcineurin.
So these are all GFP. They have a GFP construct in it. These cells.
Yeah, that's sensitive.
The 4029 is the weak though.
It should be a GFP. I don't know if it's gonna, how it's gonna relate to your stain here.
Here's your time 0.
But there's no docs. So
You don't. Well, you have.
time 0 is no docs.
Oh, there's no docs there.
Yeah.
Gotcha.
Yeah, but this was, I think, the protocol we agreed upon, and that's how we performed.
Yeah.
Amen.
The excitation emission.
Because GFP, we had to maybe do a high resolution microscopy, but calcium AM we do just to see the total distribution of cells in this 2 microliter microtumor bead. So this gives us a good indication of viability. It's not quantitative, but that's why we do MTT. So if you keep, go ahead.
Like no dose cohort for the same, cell line?
Say it again.
Do you have like a no dose a group for the same cell line?
No, we didn't do in this one, this experiment. Everything we did with the docs treated. So if you go through this, Amit, keep going through images and you'll see various cell lines. So these are all A59. I think there are 9 of them. And
wait, who's dropping out there?
That's not really
yeah, this one has, like you can see that day 21, there's a drop in growth, and which parallels what you see in the calcium imaging.
They don't have GFP. Selecta doesn't have GFP in there.
Oh, okay. Yeah, we didn't know which one is which.
That's selected.
Yeah, after we go through all these images, individual cell lines, the last slide will have all the data combined, the MTT data. So you can look everything.
Oh, 21, everyone looks kind of down.
That's too many cells.
Yeah.
And let me highlight one thing. Can you stop here? This H1703, you see the beads are sort of shrinking in size over time?
Yeah.
And that is a typical property of the cells. Unlike A54, where you did not see the shrinkage of bead, these cells may be pulling the matrix together, and that's what causes the microtumors to shrink. But there's no effect on the cell proliferation, as you see from the graph. The cells are still growing in the bead, but it's very hard to image when they start shrinking.
Yeah, the only thing that I'd point out here is that for the 1703 in this particular construct, this is not going to have a GFP. Everybody with 4029, there will be a GFP that's being co-expressed, but not with the...
not this one, okay.
Yeah.
Okay, so that's good. So this is here, all you see is the calcium AM, the live cells, but the true proliferation or the growth index you can see from only MTT assay, quantitative. So, and there are, I think, 6 H17 cell lines and 3 HT29. So HT29, as you can see, will form very nice grape-like colonies. So this is the last slide, which we have put together, and we can further replot it depending on if you want all the A49 in one group, we can put them on one slide and H17 on another. But for this discussion, we combined all the 18 cell lines and put them, the MTT graph, together from day 1, 7, 14, and 21. So without looking much, what I noticed that if you look at 2 cell lines from A549, number 4, NSD3, 1 and 2, there is a slight growth inhibition, very significant growth inhibition on day 14, but then the cells take off on day 21. So that is something to be noticed, that we saw a drop on day 14 in these 2 cell lines. And you can go back and see those original graphs. So that's one interesting point to note. If you look at the set A549 throughout, so 1, 2, 3, 4, 5, 6, 7. Almost 7 cell lines from 405, 53, 54, NT. There's a significant inhibition of growth on day 21. That's something you can notice on these growth curves. And if you look at ST29, again, there's a growth inhibition, sort of dose dependent, time dependent, on day 7, 14, and 21 in all 3 cell lines. And this is the MTT assay.
So for the drop of the, in the day 21, is that because maybe there's too many cells to start with that you kind of saturated, or just because maybe in day 21, the center core is more hypoxia, that you lose more cell? I mean, do you see this kind of like decrease in parental A549?
No. Parental cell lines will keep growing. They will not. We have done like up to 28 days ST29s.
With
the curve will keep going up. Only like after day 40, we see as like taking the growth curve just starts plateau.
Positive controls are RPA3.
But they are no. Yeah, we haven't done control in this case.
Which one?
Well, so in the 10-off version, have we seen this data yet?
No, they're doing that.
Yeah, so other set is right now under, I think we are today finishing the day 14 on the TET-off set, and that data will be available probably end of the month. And we are doing exactly same readouts, the calcium and MTT on these 4 different days. And we are following your protocol to wash out the DEX.
Yeah, I just want to go back to the last 4, the HT29, the 4029, the M1, and the M3. So just the 4029 is just the vector control itself. And it seems like there's no difference between that and the other 2, M1 and M3.
Yeah.
So again, I just want to go back to Ying's question. At 21 days, it looks like everything goes up at 7, everything goes up at day 14, but at 21, basically, everything's dying out there relative to the NT. It suggests to me that they all basically grew at a similar type of proliferation rate, but there's basically no effect here, because at 21, everybody's dying out.
Yeah, it could be, yeah, exactly.
Yeah.
You don't see, between the groups, there's no difference, but the pattern is same in all 3. Yeah.
And then I guess since we don't have a TET on, TET off comparison, if we're just looking at the NT alone for the A549, so you see A549 NT, and then you see DHX2, and looking at the purple DHX3 over that time course, again, you don't see at day 21 a big drop. You see a drop off relative to day 14, but it's whatever it is, 25% or so.
Yeah.
And then obviously, as you pointed out, the one with 4029 NSD2 and NSD1, those stand out for whatever reason. At day 14, there was not much there at all. They grew certainly at day 7 like gangbusters, but day 14 nothing, and then they suddenly started growing back like gangbusters again at 21.
Mm-hmm.
The A549s, the 2 first, the Luke and the EM4 are both controls, 2 slightly different vectors, but if you look at the 53 and 54 relative to those guys, relative to the EM4, again, you see good growth out 7 days, 14 days, and similar types of reduction at MTT assay at 21. So the MTT kind of makes sense, but I don't know how to interpret that relative to the images that we're seeing with the calcium AM.
Yeah, you're right. Calcium AM is not it's very hard to interpret that image. It just shows, like if you see a significant drop in cell number and viability, you can correlate. But the only quantitative data you'll get is from the MTT assay.
Okay, so my interpretation is like, because the control here, the neutral control here is the NT, the luciferase, and the EM4.
Mm-hmm.
So, but all those you're showing like they stop proliferating on day 14 already. Most of them. It kind of flat compared day 7 to day 14, and then you're going to see a drop on 21. So either activation of those hairpin have a toxicity. We don't know. I mean, but that's.
I would be very careful with putting too much into this. So, you know, I think this is Yvonne, by the way. This is a first glance. So MTT, what does it measure? It measures dehydrogenase activity. It's predominantly measuring mitochondrial dehydrogenase activity, but that's not necessarily, you know, the true 100% either. So you can, you know, think about it in several ways. Either, you know, it stopped proliferating, like you said. It also has maybe gotten more glycolytic, so you're losing mitochondrial function, which is to be expected, or you're starting necrotic process in the core, right? So we're doing the same type of work here for the ADC folks, you know, developing 3D models to get ready for what they want to look at. And I think this is, you know, just a very simple black and white approach of, you know, visualizing things. If you really wanted to understand more of what's happening, you need to go in with specific endpoints that either look at hypoxia, at apoptosis, at mitochondrial function, you know, through the entire sphere. So I would not, you know, spend too much time really thinking about it. I think the take-home message for me here is looking at this from an, you know, outsider, not being familiar with it. It's like I see three different patterns. You know, one is where you get that decline at 21, one where it's still the same, and, you know, one where you get something in between. So I would be not putting too much into an MTT assay at this
No, I'm not trying to make a conclusion. I'm just saying that in the dox condition, you know, all the three control in the MTT assay seem to be flat between 7 to 21 days. But we do not have a no dox control to say that is this because the MTT assay that, you know, give you a this kind of readout even in the no dox control, because you have to compare the no dox versus dox. With everything with dox, like it could be a dox effect and could be like the hairpin effect that we don't know. So basically, it's I don't think that I don't think I could draw any conclusions from this yet.
Yeah, so I guess I was agreeing with you, but I think we should move on from the MTT because otherwise we'll never get there by noon.
Sure.
That's fine.
Yeah, and this is like everybody else is saying, this was our first trial with these cell lines and testing them in the microtumor assay. So we just followed the protocol we agreed upon, and we decided to do these two analyses. So we are now open to do what should be done next. We still have the second set to be finished, and then we can look at 10 on and 10 off comparison side by side, and then we can revisit what is the next work plan. And we have some frozen microtumors, which we can provide you for some of your own analysis if you want to look at hypoxia or apoptosis or some of those using biomarkers.
Yeah.
That might be interesting.
Yeah. So you can do a lot of, the 5 microtumors will give you enough cell microDNA or RNA to do some of your gene analysis or biomarker analysis.
Now, the next ones in the 10-off vector will be more maybe interesting because they don't have the GFP, which would be potentially interfering.
Okay, okay. Yeah, I think the GFP we cannot figure out much from the calcium AM because that's a very typical non-quantitative analysis we do, because if the cells are not growing, we can see very distinction from day 1 to day 21. But as you saw, we can see the morphology that, like HT29, they made very nice colonies throughout the gel, and very unique their phenotype. And other, like your one cell line caused the microtumor gel to shrink. So that's another property we see when we do the calcium image.
The 1703.
Yeah, 17. Yeah, right.
Yeah.
So this was just a trial experiment, and like I said, we are open to you can discuss what should be done next, or we can do a very focused study or incorporate your session. And so we are open. This was the first trial experiment. So just
yeah, I think we could probably cut down on some of the constructs here based on the data. I don't think we need to run all these guys.
Exactly.
That'd be the first thing I would say. I think we definitely have to have kind of a docs, no docs, side-by-side comparison.
Okay.
And I think we can, yeah, those are the two things that jump out at me. I think we can definitely shorten the number or reduce the number of cell lines constructs, and then just make sure we run plus or minus the docs controls to make sure we're seeing specific knockdown and effects related to the gene knockdown and nothing to do with the docs induction. Yeah,
and I think, Don, you mentioned, I think earlier, that probably at day 7, we may see a little bit maybe a genotypic change, or maybe a functional change will happen maybe at a later day, like 21. So we don't know yet from this profile.
Just a quick question. We would start these experiments after the 3943 experiments, or would these be starting before those get completed? If we were to move to the next stage with these guys.
The next one, I think I will suggest let's finish. We are very close to finishing your TET-off cell lines. So by end of this month, we'll have those data.
End of April?
Yeah.
Okay.
End of April. We are finishing today, day 14.
Okay. No, all right.
So we have another week to go, and if everything goes well, then I think in 10 days or so, I will have the similar report prepared. Okay. And then Don can share that with you guys. And so I think you look at these data and give us the feedback, and let's look at the project B, which is the TET-off cell lines. And then I agree, like we select the cell lines which are of interest and do a detailed analysis.
Okay. Are you going to send slides out to everyone?
Yeah, I think, Don.
I will do that, Raj. This is Amit.
So why don't you just send this PDF to everybody? And so any questions or any information you need, you can send an email on a technical side. If you have any, I'll be happy to answer that.
Yeah, I actually had a number of questions written down. I just got back to that as away for 2 weeks, but yeah, maybe I'll send some of those questions as well. They're more general questions than specifically related to this particular experiment.
Yeah. Okay. So protocol is very general. We didn't put a lot of details, but if you have any clarification, I'll be happy to provide.
Okay.
And can you just, when you send those questions in, can you just make sure you copy myself and Amit so we can help coordinate and make sure everybody's, that information is included in the portal?
Yeah, I will definitely do that.
Yeah, we are sending everything to Don, so it's shared across everybody, so everybody knows what's going on.
Okay.
And one other question, when should we book the next time to talk, Raj? What would be a good?
I will say first week of May, because I'll be gone after that for at least for 2 weeks. So
and
early May.
How does everybody look for beginning of May?
The first week could be good.
Yeah, first week, like May 5th week of that.
Do we want to do the same day, same time, or do we want to do Tuesday to give us a day to get back?
Tuesday.
I
do Monday
either way. Yeah.
Sorry, what did you just say?
So Lisa and I can do Monday or Tuesday. Tuesday is better. Yeah.
Tuesday same time, maybe Dawn.
Yeah.
Okay, sounds great.
Yeah, and by then we'll have the report of the second set.
I have one more quick question, if you don't mind.
Yeah.
Okay. I'm just thinking about, where I sit, where I have to kind of look at reproducibility, and I know, how difficult it is for us just, to get the normal 2D into something reasonable. And so again, we're working on 3D here as well. So I noticed that your standard deviations are actually quite lovely for a lot of the cell lines, but it's obvious that for some, like 4053 or at the end 4029, that there's rather large. So is the morphology of these cells quite different?
No, I think it is the problem with the microbeads sometimes. There is an outlier, like these are, I think, average of at least 5 microtumors for each. So most of the time they are pretty tight, but what happens, like when we are doing MTT assay, we have to solubilize the whole bead.
Right.
And sometimes, so sometimes all the substrate doesn't come out, or there is some variability with 1 bead or 2. Yeah.
So, but do you think it's just that, or do you think there's something specifically to the cells? Because, when I look here at the cells we work with, and we have like 5 or 6, they're very different in their behavior. So I could see, something that looks like a fried egg being differently associated with the matrix than, something that's nice and tight. And so I'm just wondering if there's something, and I guess I can ask the question to Dawn as well, or Andrea.
Yeah, these standard deviations can be easily addressed because, like I said, we can run a few more beats and throw out the outliers, those kind of things.
So, Dawn or Andreas, do you know anything about those, or is it, would you look at the standard deviation and say, oh yeah, those are those cells, yeah, good heavens, or is it? Yeah,
these cells we have used a lot for various projects, so we haven't seen many problems. Like only when the beads are shrinking so much, and you cannot get all the, the MTT readout, then there could be a problem.
Okay, thank you.
Yeah, because if the cells are pulling the matrix and they become so small, they will basically suffocate cells. So that could be a poor readout for those cell lines. And we saw that some of that effect over time with one of the cell lines. So we can maybe look carefully that these MTT data fits pretty tight with the readout.
So Raj, that's sort of your perspective on Yvonne's question, but I think she was also looking to get some of her own people to comment on the question.
Sure.
So just to see if, I think it was Andrea or Dawn, do you have any comments on Yvonne's question?
Well, I'm looking at those standard deviations. I mean, some of them at 7 days. If you're looking at the 40, A549, 40, 54, they're actually quite tight. The 21-day or the 14-day seems to be the outlier there. It seems to be a lot higher in some of those. But again, if you just, I'm looking at, try to look at them in terms of different sets. The first set with just the A549 DHX9 guys, they look pretty similar. The next set with NSD3, again, they do kind of go up and down a little bit because those day 14s drop out. I don't know what to say about that. And then you get the 2 controls for A549, and then the 40, 53, and 54. Those kind of jump out at you. Those standard deviations look very high, and they look very similar to the HT29 M1, M3s, which relative to the NTs, they look like they're a lot higher. I guess that's what I would say, but maybe to Raj's point that if you put a couple more wells or microtumors in there, some of that noise would come out. So,
yeah, we have seen this, and like next time when we do, we can always add more N numbers. So you'll get a very tight ST.
Yeah. Yeah.
So, still, yeah, some of those, I think, questions or suggestions, just put in your...
Raj, how did we came up with the MTT? I mean, is that did you guys use that before? That's how we went with the MTT assay instead of, let's say, a cell titer glow?
Well, we use either ATP light or MTT in here in our lab. We don't use cell titer glow, but if you want that, we can do it.
Because I mean, what we've seen from our assays when we do the 3D, it seems like the cell titer glow will give you a bigger range, so you will have, maybe your standard deviations are not as, yeah, to me, like, they look a little high, but the cell titer glow, because of the range, you could really see if you have, you know, because of the spectrum.
Yeah.
So, I mean, I don't know really if it was, you know, we already decided that, you know, with the MTT, but we're trying to move away also from the MTT because of some of these issues.
One thing we consider the cost. The cell titer glow is a lot more expensive.
Nah, I mean,
that's one thing.
Not really. I don't know. I mean, I don't know what you guys, but for us, I mean, it seems like maybe a little less.
Oh, really? Okay, maybe I'm mistaken. Okay, but I'll check in lab. Means if you want, we can run it. It's not a very difficult thing for us to change. But like during discussion,
because you lyse the cells with the cell titer glow, you completely lyse the cells, so you could, and it's much quicker, because with the MTT, I just don't know, like, if you're getting, you know.
We lyse we do the lysis in that set too. But like I said, the cells are growing inside the gel, and that's where the problem is. If you do not fully lyse the bead, you will have the variations.
So how do you, I mean, with the MTT, you lyse them?
Yeah.
Yeah, it's just an enzymatic assay, so yeah, of course you lyse.
I think you're, again, what I see there, for a lot of the lines and a lot of conditions, the standard deviations aren't that bad. So there, you're at least getting relative. I mean, we don't know if the number of cells are different in those microtumors because you're only taking a couple of those guys. But it doesn't look that bad. I think the point that I would agree with Andreas is that maybe the dynamic range of the cell titer glow assay might be a bit better readout than the MTT moving forward.
Yeah, right.
Like
in the protocol, it said like we add 10% SDS, and it goes overnight, and the bead is completely dissolved. So that's what we do it overnight.
Yeah. So.
But these standard deviations we have seen, like they are very easily adjustable. It's not that you can get a lot tighter points if you do more numbers of microtumors per assay.
Okay.
Yeah. But as you see, like most of the time, we get pretty good average from 3 or 4 microtumors. And since there are so many cell lines, means we just kept everything to minimum. So.
Well, I think it'll be interesting, as Max was alluding to earlier, to see what the 3943 looks like. We won't have any GFP there,
okay.
And I think
we've got 2 of our best positive controls.
Yeah, we have 2 really good positive controls in there. So I think just in terms of a data set, it should be a little bit more enlightening in terms of the activity and the consistency across the different readouts. That'll be very helpful. And I think from there, in looking at this set here, we can craft a smaller set of cell lines and probably a better set of assay readouts to follow up with a stage 2 then.
Right, yeah. So I think I agree that you pick the cell lines you think will be interesting to look at again, and we do this, repeat the same experiments with and without dogs, and have the controls positive and negative.
Oh, we've got some good ones too, some new ones.
But this is the first shot, so it's not that bad. So.
Okay. Well, we'll craft some questions here. Andreas, Ken, we can come down and chat with you guys too, and do some other thoughts and points you want to bring into this. And we'll put together some details for the next round. Send it back to you guys, and we'll look forward to talking again in about 10 or 11 days then.
Yeah, and by then I think we'll finish the second report, and Don, I think we can circulate that, so during our next conference call, everybody can go through and have maybe some feedback ready for us.
Mm-hmm.
Sure. And the only other thing I would just remind everybody about, because I know Yvonne knows this really well, but the rest of the team probably doesn't. We typically like to find pieces of these projects that we can share with the rest of the consortium. And typically we actually do a project in conjunction with the consortium working all together, but because you have some proprietary information here, we're doing it sort of as a one-off. So I know Yvonne and I can talk about this separately, but if you can just also think about what you think at some point we can share with the rest of the consortium, that would be great.
All right. Well, duly noted. Thank you.
Who's in the consortium?
Other pharmaceutical companies like Amgen, J&J, Lundbeck, Sanofi, Infinity.
So it can be something very generic at the end. So, you know, it could be something like where we say, you know, out of 28 cell lines we tested, we find that, you know, the best time for harvest in this case was 14 days or 21, and these are the endpoints so you don't have to, you know, give anything away. It's more a general learning.
Yeah, understood. Okay.
Yeah, so we look forward to your feedback and suggestion, and just like you said, incorporate anything you can come up with, and we look at the second report, and then we reconnect in early May and plan out our next round of study.
All right. Thank you so much for the data and the time and effort to put that together then.
You're welcome.
All right. Take care.
Thanks. Bye-bye.
Bye-bye. Thanks.
All right, guys.
Have a good day. Bye.
Bye-bye.